Good afternoon, everyone. Thank you so much for joining us today. My name is Emily Silva, and I'm the NECAN coordinator. I'd like to welcome you to the fourth webinar in the NECAN Student and Early Career Scientist series, in which we are highlighting the work of students, PhD candidates, and early career scientists who are working on ocean acidification. The full schedule, along with past webinar recordings, can be found on the NECAN website, necan.org/student-webinars. And also, don't forget to sign up for our newsletter so you don't miss any future announcements. Throughout the presentation, if you have any questions, you can submit them through the questions tab on your GoToWebinar control panel, and we'll go through each of those during the Q&A session following the presentation. Today, we'll be hearing from Louise Cameron from Woods Hole Oceanographic Institution on the consequences of ocean warming and acidification for the Atlantic sea scallop fishery. Louise is a marine carbonate chemist and ecologist whose work focuses on the impacts of climate change on bivalves and their fisheries. She's originally from Scotland and moved to the US to pursue her PhD at Northeastern University. She's now a postdoc at Woods Hole Oceanographic Institution, where she's modeling the carbonate chemistry of the Northwest Atlantic shelf to help the Atlantic sea scallop fishery prepare for ocean acidification. So with that, I'll hand things over to Louise and she can take you through her presentation.
Okay, hi everyone, thanks so much for joining today. So my research focuses on predicting the consequences of ocean acidification for the Atlantic sea scallop fishery. I started my postdoc this year, so I'm mainly going to be talking about the results from my PhD research at Northeastern, but I'm also hoping to get a little bit into some of my postdoc work closer to the end of this presentation. So ocean acidification occurs when carbon dioxide dissolves in our surface oceans. Once it dissolves, it reduces both pH and carbonate saturation state. Because carbon dioxide is more soluble in colder waters, ocean acidification is happening at a faster rate in high latitude regions when compared to the tropics, for example. So I'm currently based in the Northwest Atlantic, and as you'll see, this region is expected to experience a decline in 60 to 65% carbonate saturation state from pre-industrial times by the end of the 21st century. At the same time, some reports also suggest that this region might experience a more rapid rate of warming compared to the global ocean average. Now, this region is also a habitat for a lot of commercially important bivalve species, which have been shown to be particularly vulnerable to ocean acidification in meta-analyses. These bivalve species are a really big source of income for this region. So this data that I have here is from financial data from 2018. And these 4 species combined generated more than $450 million for the local economy in this year. So it's really important that we quantify the effects of ocean acidification on these species. So my dissertation research focused on the response of the Atlantic sea scallop to both ocean acidification and warming. And I asked this question in a number of ways. So first of all, I did a field study where I measured scallop performance across a natural carbonate chemistry gradient. I also carried out a mesocosm experiment where I measured the response of Atlantic sea scallops to both experimental ocean acidification and warming. And currently the work that I'm doing at Woods Hole is expanding on this research to simulate the effects of future ocean acidification onto the Atlantic sea scallop fishery. So a large portion of the Atlantic sea scallop fishery takes place on Georges Bank, and this region is really oceanographically interesting. It has a large depth gradient. It also experiences upwelling, it has strong tidal currents, and it has a wide variety of sediment types, and all of these different factors might lead to a high level of heterogeneity in benthic carbonate chemistry across this region. However, Georges Bank is a bit of a data gap, and there are very few measurements of benthic carbonate chemistry across Georges Bank. As I mentioned, this region is important for the Atlantic sea scallop fishery. So Atlantic sea scallops are found in stable aggregations across Georges Bank. In this figure, you can see stable sea scallop aggregations shown here in yellow and brown. And the scallop fishery is managed using a rotational management strategy. So fishing areas are either closed to allow the population to grow or open to allow fishing depending on the state of the stock. One of these areas, the Nantucket Lightship area, has experienced mass mortality events in recent years. And the scallops here are also known as Peter Pan scallops because they're simply not growing very large. So I was first interested in using this region as a kind of test bed to look at natural differences in carbonate chemistry across Georges Bank. I then wanted to answer whether seawater carbonate chemistry affects the meat and shell conditions of Atlantic sea scallops. And I was also interested in whether seawater carbonate chemistry affects Atlantic sea scallop growth rate. So to answer these questions, I went on a 5-day field cruise on Georges Bank aboard a commercial fishing vessel, the FB Liberty. And we visited 15 sites across Georges Bank. So the white dots on this map here are my field sites. You can see the tip of Cape Cod in the top left corner here to kind of orient you to where Georges Bank is. And at each of these sites, I collected 2 water samples. And then I took them back to the lab and measured the dissolved inorganic carbon, alkalinity. I measured the temperature in the field, and I also measured the salinity of these samples. And I used this to calculate the entire carbonate system. But the aspect of the carbonate system that I was most interested in was the calcite saturation state. This is because calcite is the form of calcium carbonate that scallops use to build their shells. So the data that I report here on carbonate chemistry is all going to be saturation state. So on my map now, you'll see that there are some numbers associated with each of my field sites. This is the measured bottom water saturation state at each of these sites. Now, to give a quick introduction to saturation state, when saturation state, or omega, is greater than 1, this supports calcification. So under these conditions, we would expect shells to be able to readily grow. When saturation state is less than 1, we call this the dissolution threshold. So when saturation state is less than 1, we would expect shell material to actively dissolve in seawater if there were no other, for example, biological factors influencing or protecting those shells. So you'll notice that some of my sites had a pretty low saturation state. In fact, one of them crossed the saturation boundary into the dissolution region. But I did have some other sites that had pretty high saturation state as well. So I found that there is a high level of variability in bottom water carbonate chemistry across Georges Bank. Now, the next thing that I did was collect some scallops. So at each of my field sites, I collected scallops using a commercial dredge. We towed this behind the boat for 15 minutes at a time. The dredge was lined with a mesh to capture scallops of all size classes. We then emptied the tow onto the boat. This was actually our largest tow and contained about $10,000 worth of Atlantic sea scallops just in this one tow. I then quartered the tow again and again until I just had 50 scallops left to create a random sample and brought these back to the lab. At the lab, I dissected my scallops and measured the dry weight of all of their tissues to allow me to calculate different tissue indices. So the first one that I'm going to talk about is relative tissue condition, which is the dry weight of all of the tissues divided by the allometrically scaled shell height, so the height of that shell. And because obviously we're in a field setting, so there are a number of different things going on, as well as calcite saturation state changing, we also have depth changing and other environmental factors changing as well. So I conducted several different linear mixed effects models, and then I used the AIC criteria of these models to select the model that best explained my data. I did this with all of my field results. So in the case of relative condition index, I found that calcite saturation state best explained my data. So just to orient you to the axes of my graph, I am going to have calcite saturation state on the x-axis. Now remember that when saturation state is low, pH is also low. So we're getting into more ocean acidification-like territory the further left we move on this graph. And then on my y-axis in this plot, I'm going to have relative condition index. And so I found that there was a decline in relative condition index in response to lowering calcite saturation state. So this means that under future ocean acidification, we might expect scallops to produce a lower amount of tissue. So the next index that I was interested in measuring was gonadosematic index. This is the weight of the gonad tissue compared to the rest of the tissues. And this gives a measure of the reproductive health of scallops. And again, I found that this was impacted by calcite saturation state, and again, this declined as saturation state got lower. So I was also interested in the effects of carbonate chemistry on shell properties. And this is because the shell is the scallop's main defense against predators. So predators like starfish access scallops. They need to be able to pry apart these shells. Some predators will also chip away at the edges of the shells, like crustacean predators. And so one way that scallops can protect themselves is by growing strong shells. So to quantify shell condition, I measured shell strength and shell microstructure. I also was interested in shell growth. So another way that scallops can protect themselves from predators is by reaching what we call a size refuge. So this means that they grow large enough that a predator is no longer able to access them because they're too large. So to measure shell strength, I used an Instron materials testing machine, which is essentially a giant shell crushing machine. So I put my shells in this chamber, and I had a bar that got pressed along the midline of each scallop shell. And with this bar, I applied increasing force until we reached a catastrophic breakage point. And then I recorded the force that it took to break the shell. To quantify shell microstructure, I used a scanning electron microscope, and this allowed me to take really high resolution images of the inside surface of those shells. So this is an image of a scallop shell magnified 15,000 times. And this allows us to see all of those individual calcite crystals. So all of these crystals that you see here, these are the individual calcite crystals that form the shell. So in each of my images, I placed a box of known size over a random area in the image and counted all of the crystals inside this box. And I did this 3 times in 3 random areas on each image so that I could get an average crystal distribution density. So let's take a closer look at these images. So this first image is from one of my high saturation state sites. And you'll notice that those crystals all have a very regular shape and size. They're all oriented in the same way, and they're overlapping quite a lot. When saturation state gets lower, you can see that we still have that regularity in shape and size of the crystals, but if you look closely, you'll see that there are some gaps starting to appear between some crystals. We also see the crystals look a little bit longer. This just means that they're moving apart a bit, so more of each individual crystal is visible. When saturation state is lower still, we start to lose some of that regularity. So you'll see some wider crystals. You'll also see some long, skinny crystals. Again, all of these crystals generally look longer. So we're seeing more of each individual crystal, showing that they're less densely packed. And at my lowest saturation state site, I couldn't even quantify crystal density because the edges of these crystals showed such strong signs of dissolution that it wasn't possible to quantify what a crystal was. But I found that calcite saturation state impacted crystal distribution density. And I also found that shell strength was best predicted by a model that included both crystal density and calcite saturation state. So in this figure, I have crystal distribution density on my x-axis, and I have catastrophic breakage force, or the shell strength, on my y-axis. And when my data points appear, they're going to be colored according to calcite saturation state, with high saturation state in red and low saturation state in blue. And so as you can see, shell strength is declining as distribution density gets lower, but also as saturation state gets lower. So finally, I wanted to understand the impact or the effect of carbonate chemistry on growth rate. So I had to age my scallops. Now, one way that we can age scallops is to count the annual bands that are on the outside of their shells. So just like tree rings or coral skeletons, scallops lay down annual growth rings, which allow us to count them and then estimate their age. But scallop shells are pretty messy compared to some other species, and they tend to have things like shock rings. So if you look at this figure, we can see shock rings pointed out by arrow C, which can get confused with the actual annual growth bands, which are shown here in arrow D. So instead of looking at growth bands in the shell, I looked at growth bands in the scallop's rhizolium. So this is a calcareous plate that sits underneath the protein hinge that joins those two shells together. And when we scrape away the protein component, underneath we see these really distinct annual bands, and we don't get the shock bands shown here that we see on the shell. So this makes it easier to more accurately quantify scallop age. So in this graph, I'm going to show a growth curve. So I'm going to have estimated scallop age from those rhizolium bands on the x-axis, and I'm going to have the scallop shell height on the y-axis. Again, my data points are going to be colored according to calcite saturation state, with high saturation state in red and low saturation state in blue. And I found that growth rate was best explained by a model that included calcite saturation state, where the lower the saturation state, the slower the growth rate. So this means that scallops might not be able to reach that size refuge as fast under ocean acidification, but it also might leave them, mean that it takes longer for them to enter the fishery as well. So to sum up what I found in my field survey, I found that sites with low saturation states had lower meat condition. This suggests that under future ocean acidification, we might see a transition from these large, chunky, profitable meats that we see on the left here to scallops with smaller, less profitable meats, as we see on the right. I also found that gonadosematic index was lower under low saturation state. This might mean that scallops aren't able to put as much energy into reproduction in the future, and we might eventually see a smaller scallop population because of this. I also found that shells were weaker under low saturation state, which might mean that we see higher predation mortality under ocean acidification. And I found that scallop growth rate was lower under low saturation state, which again could result in increased predation mortality and could also mean that it takes scallops a longer time to reach a profitable size. And I also find this high natural variability in carbonate chemistry across Georges Bank. Now, everything that I've talked about so far was measured in the field, and when we're working in the field, there's so many other factors going on that we can't control. So everything that we see in the field is, we can say that there are correlations, we can't say that there is causation. So to understand whether the changes in carbonate saturation states were what were causing the effects that I saw in the field, I conducted another study, this time in a laboratory setting. So I did this experiment in the ocean acidification array in the Reese lab at Northeastern University. And in each of these tanks, I can adjust the bubbling rate of carbon dioxide and air into these tanks to change the carbon dioxide concentration, and I can also change the temperature of the tanks. So for my experiment, I had a control saturation state of 3. I had a moderate ocean acidification saturation state of 1.6. And I had a high ocean acidification treatment, which was just at or below that dissolution threshold. And I did this experiment at 3 temperatures, which were 6 degrees Celsius, 9 degrees Celsius, and 12 degrees Celsius. And in each of my treatments, I had 3 replica tanks, which each contained 6 scallops. And I carried this experiment out for 4 months. So the first question I was interested in were whether ocean acidification and warming affect Atlantic sea scallop growth rates. And so to answer this question, I used buoyant weights. This is a technique where we essentially put that scallop on a platform and hang that platform from a bottom-loaded scale. This platform is submerged in seawater, and this means that we only get the weight of the shell because the shell is negatively buoyant, whereas the tissue is neutrally buoyant. I measured buoyant weights once a month, and I held the water bath at constant temperature and salinity in each treatment in each month, and this meant that this wasn't impacted by any buoyancy effects. And then at the end of the experiment, I carried out a dry shell weight to buoyant shell weight regression to make sure that I was getting the actual change in calcium carbonate material over the course of the experimental period. So I'm going to show you the results of my buoyant weight measurements in this figure. And again, I'll start by just orienting you to my axes a bit. So I have calcite saturation state on the x-axis. And again, remember that as this gets lower, we're getting into more ocean acidification-like territory. And then I have calcification rate on my y-axis. I'm going to have a dashed line at 0. Everything above this line is net growth, so those shells were growing throughout my experiment. Everything below this line is net dissolution, so we saw those shells losing weight. And then I'm going to have my 3 different temperature treatments in 3 different colors. So my lowest temperature treatment is going to be in blue, my moderate temperature treatment is going to be in orange, and then my high temperature treatment is going to be in red. So as you can see, in all of my temperature treatments, calcification rate declined under ocean acidification. But what was interesting to me was that we actually saw an interaction between ocean acidification and warming. So if we look at my, control calcite saturation state, warming had a positive effect in this treatment. So my growth rates were fastest in the warmest treatments, whereas at my most acidic treatment, warming had a negative effect. So the next thing that I looked at in this experiment was whether ocean acidification and warming affect Atlantic sea scallop survivorship, because I did see some mortality in my experiments. So these graphs are showing the results of a proportional hazards model. I'm going to have time in days on the x-axis, and I'm going to have estimated survival proportion on the y-axis. So that's the number of scallops that were alive at a given day in the experiment. And again, I have my different temperature treatments shown in 3 different colors, with blue being my lowest temperature, amber being my middle temperature, and red being my high temperature. So this first graph here shows my control ocean acidification treatment. And as you can see, in my lowest temperature treatment, I really didn't see much mortality at all. So most of the scallops survived until the end of the experiment. As temperature increased, however, we saw an increase in mortality. The same trend was shown in my moderate ocean acidification treatment. And I also found the same thing in my high ocean acidification scenario. Now, if we look across these graphs, you'll see that the slope of the decline in survivorship over the experimental period is increasing. So what I found was that there was an additive effect of both ocean acidification and warming on survivorship. So to sum up what I found in my field study, I found that in my tank experiment, I found that calcification rates declined under ocean acidification, and this really kind of corroborates what I saw in the field. I also found that calcification rates were impacted by temperature. I found that global change impacted survivorship. So again, this might cause the scallop population to decline in the future if what we see in the tank experiment is realistic to what we see in the field. And I found that multiple stressors have both additive and interactive effects. So because ocean acidification and warming are going to co-occur in this region, I think it's really important to study their combined effects. So currently, I'm looking at spatial variability and ocean acidification across Atlantic sea scallop fishing grounds. And this work was really inspired by that finding of high heterogeneity in bottom water calcium carbonate across this region. So one thing that I can use to look at differences in carbonate saturation state across this region is historic data. So I looked through the archive of historic bottle samples that exist. And as you can see, there's not a ton of these, and most of the sampling has taken place within the last 5 to 10 years. The samples that I show here are also across all depths. So what you can't really see in this image is just how few exist in bottom water, which is where the scallops live. However, one thing that we do have a lot of is CTD casts, and these of course go right down to the bottom water as well. So right now I'm involved in a project that uses these benthic bottle samples to train machine learning algorithms to understand what other data sets we can use to predict benthic carbonate chemistry. So in my bottle samples, I'm using the salinity, the temperature, and the sample depth. I'm also collecting data on bathymetry and the distance of each sample from the bottom from bathymetric data sets. And then I'm using CTD casts to calculate the mixed layer depth and to calculate sea surface temperature. And then I'm using satellite data to calculate or to estimate chlorophyll A concentration and also sea surface height. And so all of this data is then associated with the date and time and location of each of those bottle samples. And then I'm using different machine learning techniques to understand which of these variables can be used to predict the dissolved inorganic carbon, or the dissolved inorganic carbon, sorry, and the alkalinity of my water samples. So this first model I've conducted, this is very preliminary data, and I created this model when I only had access to a subset of these variables, so hopefully we'll get increased accuracy the more variables we add. But as you can see, there's a correlation between the measured dissolved inorganic carbon in the bottle samples and the predicted dissolved inorganic carbon that we predicted from the models. So this project is in close collaboration with Jenny Rubin and Ivan Lima, who both also work at WHOI. And then this summer, I'm hoping to fill in some of those data gaps on Georges Bank. So in May, I'm going to be doing a carbonate chemistry cruise across Georges Bank. So each of these shaded regions on this map are areas that are under management by the sea scallop fishery. They're included in the SAMS fishery models for this species. And then I'm going to pull up the sites that we plan to visit. In black, you'll see sites with pre-existing data, and in red, you'll see sites that we plan to tow for scallops to create or to generate more data on linkages between carbonate saturation state and scallop meat and shell condition. And so we're planning to visit about 114 sites across the bank. We also decided to throw in some sites kind of in the center of the bank there to look more at the oceanography of this really interesting region. And then once this data is collected, it will be fed into an integrated assessment model that was created by several scientists, including Jenny Rubin, Sarah Cooley, and Devorah Hart. And Alec Wang is also going to be a collaborator on this model as well. And so the data from this field survey, as well as all of the historic data that we are able to collect, is going to be fed into this biogeochemical submodel. So we'll divide this model up into the different scallop management regions. This will then feed into a scallop submodel. And I'm going to be using data from my tank and field studies to kind of hone some of the parameters in that model based on what we saw in these studies. And this will then feed into a socioeconomic model to predict the effects of ocean acidification on future sea scallop stocks. So I feel like I went through all of that pretty fast, so I've definitely got time for acknowledgements. So I'd like to thank everybody who's helped me on this research. So I want to thank my, my supervisors at Northeastern University, John Grabowski and Justin Rees, my collaborators and supervisors at WHOI, Alec Wong, Jenny Rubin, and Ivan Lima, all of the undergraduate assistants who helped me both in the lab and in the field, my wonderful lab mates both at Northeastern and now at WHOI. And particularly I want to thank the field crew who came out and helped me in the field. So this were the fishermen at Nordic Inc. And I would also like to thank my funding sources. And I'll happily take any questions. Thank you.
Thanks, Louise. That was a really great presentation. We do have a question that has come in. They ask, is there any data with sea scallops or other bivalves on the effects of OA on the composition of the extrapallial fluid from which calcification takes place?
Darn, I actually took that out of this talk because I thought I'd go over time. Yeah, actually, a big part of my thesis was looking at extrapallial fluid chemistry. So I measured the pH and the dissolved inorganic carbon and the salinity at the site of calcification. And I found that scallops regulate both the pH, the alkalinity, and the dissolved inorganic carbon at their site of calcification to kind of try and create conditions that should promote calcification under ocean acidification. What I found, however, was that even though they were able to create conditions that should be quite good for calcification in their extrapalaeal fluid, which is the fluid that they build their shells from, for people who aren't as familiar with what that term is, I found that this then didn't translate to increased calcification function. And I think that this could be for a couple of different reasons. They were, we saw such strong effects of shell dissolution, so perhaps even though they're building shell inside this fluid, it's dissolving at a faster rate from the outside. Another result that I didn't show here is I saw reduced respiration rate under ocean acidification, so they might not have enough metabolic energy to convert materials in their extrapalaeal fluid into shell under ocean acidification. So yes, I have a lot of thoughts about that.
Okay, the next question asks, were your SEM images taken in cross-section?
No, they weren't. Those were actually, so I chose to look at, when I say the inner surface, I don't mean like the interior shell, like it wasn't a cross-section. Instead, I mean not the surface that faces the seawater, but the surface of the shell that faces, the pearly kind of surface that faces the tissue instead. The reason why I chose this surface is because, well, one, that's where the shell is created. So we're seeing more newly formed shell. Also on the outside of the shells, they have, sometimes they'll have encrusting epiphytes, or sometimes they'll have, damage from dredges or predation or boring activity. So I didn't want any of those factors to interfere with the effect that I was trying to measure. So they're the inside surface of that shell, the side that is in contact with the tissue.
Okay, the next one says, great presentation. I guess you'll be making measurements. I guess you'll be making measurements to determine whether the low saturation state at that one site is persistent. If so, are you saying that we're already seeing impacts of OCA now and not just in the future?
Yeah, I mean, I'm definitely going, so those measurements were taken at one point in time. I collected them in August, which saturation state tends to decline when temperature gets lower. So we might actually expect to see that to get lower in the winter months, potentially. But because that was just measured at one snapshot in time, we don't really have a way to tell whether that persists throughout the growing season, or whether we just happen to catch it on that day. I do think that it probably does persist because we saw such strong correlations with scallop, both tissue condition, which can be more transient, but also shell growth. Like we saw lower growth rates at those time, and growth rate is of course capturing something that occurs over the scallops for a whole lifetime. So I think that this probably does persist over time. I am going to revisit these sites to see if we still see those low saturation states. But yeah, I think we could say that we're already maybe seeing the effects of ocean acidification in this region.
Okay. The next one says, really fantastic work. Thanks for advancing it. I'm curious if you've considered including oxygen data in machine learning algorithms. There should be much more of that than carbonate chemistry. It goes on, also please know that the NOAA OAP will be sponsoring the East Coast OA survey next summer, which will be transecting the bank. It might be good to coordinate with the UNH team leading this.
Yeah, I mean, definitely. That sounds like we should definitely talk about that because it's definitely great to get more data in this region. Yeah, we considered oxygen. However, we're only using samples that are at the exact date, time, location, and depth of our samples, and so a lot of there's a lot of oxygen data, but it didn't necessarily overlap with where we had carbonate chemistry samples for the training data set. So when we included oxygen in our models, it really lowered the amount of data that we had. There's some surveys that always collect oxygen alongside their carbonate chemistry, but not all of them do, so that didn't give us as much data. But oxygen is definitely something that could affect the carbonate system as well.
Did chlorophyll or phytoplankton concentrations vary across Georges Bank, perhaps affecting shell strength?
Yeah, that's still TBD. So my collaborator Ivan has done a lot of work downloading all of the satellite data. We just got that a couple weeks ago, and I've not really had time to dig into it yet. But stay tuned for that.
Are you using omega for aragonite or calcite in your comparisons?
In this presentation, I showed calcite saturation state. Yeah, I'm collecting data on aragonite saturation. You know, when I report this data, I'm going to show both calcite and aragonite because aragonite is so useful for other species and could also be useful. Scallop larvae, for example, use aragonite rather than calcite. But for the adult scallops, I chose to focus on calcite because that's what they use to build their shells.
When you showed the empirical fit to DIC, was that the training data fit or evaluation data set?
That was the evaluation data set, yeah.
Okay. I'm going to give you guys another minute. That was the last question that has come in, but we have plenty of time if anybody's currently typing. I don't want to rush you. So we'll just wait another minute.
Yeah, if anyone thinks of any questions later as well, definitely feel free to reach out. I think this presentation is going to be uploaded to NECAN's website, and my email is on the first page.
Definitely, yeah, this should be available on the NECAN website in the next day or two. So definitely check that out. Okay. Well, that looks like it's going to be our last question. So I would like to thank everybody for attending today. And thank you, Louise, for that presentation. Our next scheduled webinar is on Tuesday, April 20th at 1 p.m. The registration link, the title, and abstract for that talk will be shared on both our NECAN website and through our mailing list. So make sure you're on the NECAN mailing list to get those updates. I've also been posting on the Ocean Acidification Information Exchange, and I encourage everyone to request an account there if you haven't already. It's a great place to get more information on OA, to stay informed on webinars and events, and engage in conversation. So thank you again, Louise, and thank you everybody else.